everybody. Welcome to today's seminar. We have heard a lot about the promise of using EEG as a biomarker in KCNT1 related epilepsy, as well as in other DEEs. And today, we're really excited to have Dr. Peter Gaylor present on the work he's done. Dr. Gaylor earned his PhD in bioengineering at the University of Pennsylvania, where he worked with Drs. Brian Litt in the Center of Neuroengineering and Therapeutics and Ingo Helbig at CHOP to develop novel methods to extract and quantify biomarkers from multimodal data in patients with genetic epilepsies, as well as a pipeline to extract and analyze EEG data to find quantitative EEG biomarkers in rare genetic epilepsies. Dr. Gaylor currently works as a research scientist at Neumarker, whose work focuses on the discovery of novel EEG biomarkers and endpoints in neurological and psychiatric conditions. Today, he will present his preliminary findings in isolating novel EEG biomarkers in KCNT1-related epilepsy, leveraging machine learning, signal processing, and network neuroscience. Thanks, Peter. Take it away.
Great. So let me just share my screen here, make sure you can see everything. Great. So thank you for the introduction. As you've heard, my name is Peter Gaylor, and I'm very honored to be here with you all to present some of my work on quantitative EEG biomarkers in KCNT1-related epilepsy. As you know, I recently completed my PhD and then a brief postdoc, continuing my work, which I found really exciting in scalp EEG biomarkers in pediatric genetic epilepsies. And since then, I've joined a small company called Neumarker to continue this exciting work in EEG and precision medicine. And before I start, I'd also like to mention that much of this work is unpublished and results from years of work. So if you're interested in collaborating or have further questions, please just email me or Dr. Ingo Helbig. And of course, none of this work would be possible without my incredible colleagues and friends at the Helbig Lab at the Children's Hospital of Philadelphia, see on the left, and at the Center for Neuroengineering and Therapeutics at the University of Pennsylvania, seen on the right. Now, much of what I'm presenting here is based on pipelines and techniques from this paper, which I first authored. The preprint is available online for free, and the paper itself is currently under revision, so hopefully sometime soon you'll be seeing it. And of course, none of this work would be possible without my incredible mentors, as seen above from both CHOP and University of Pennsylvania. My only conflict is, of course, I'm now an employee of Neumarker, continuing a lot of my research on scalp EEG, while also continuing some of my research at CHOP. I also wish I could present some of my work at Neumarker on scalp EEG prediction algorithms, as I think it's very exciting, but hopefully sometime soon I'll be able to present it. Now, my research has focused on characterizing and predicting genetic epilepsies across development using multimodal data with special focus on disorders such as KCNT1. Here are some key points I want to get across during my presentation. Now, due to time, I'll only be presenting a small portion of my work, but I'm happy to share some other findings with anyone interested after the talk or if there's particular questions, and I'll be hinting at that at various times during the presentation. So, a bit of brief background. What is EEG, or an electroencephalogram? Well, it attempts to measure electrical activity in the brain due to complex neuronal signals. It does this by applying electrodes to the scalp to try to capture these signals. It's a bit messy, but we can begin to sort through the noise. Here is a picture that we see on the right, is what it looks like in the clinic at CHOP. This is a child with a pathogenic variant in SCN2A. Now, we've long been aware that certain disorders have visually identifiable signatures in their EEG. This typically requires years of training to learn to identify. EEG, as I've said, is a very complex signal filled with noise and artifacts, but there are patterns that clinicians can visually identify. Here are some examples that you may have heard of. These are very specific things a clinician might be able to see in an EEG and the consequent syndrome it could indicate. Many of these signatures, however, are not constant, and I'm sure many in this Zoom room are aware of getting multiple EEGs or an EEG lasting days with still no clear answers. Furthermore, they're prone to error. With EEG analyzed by trained clinicians, individuals without epilepsy have a 30% chance of a misdiagnosis of epilepsy. Similarly, expert inter-rater reliability in distinguishing epileptiform discharges or epilepsy-like activity in EEG is about 50%. Finally, most of the signal is simply hidden to the human eye. So let's take a look at an example of what a clinician actually might see in a clinical scalp EEG captured from CHOP. This is a 15-second clip of an EEG from an 11-month-old infant with a genetic disorder. It's filled with noise and artifact. On the y-axis, we see the different electrodes that were placed on the scalp. Luckily, with modern techniques and software, we can clean it up. Here's what it looks like after a pipeline I built to clean the EEG. Now it's much better, but still, what can we get from this data? As we've seen, even with a trained clinician, there's only so much you can pick up. There's rich information hidden within this signal that can be extracted computationally with quantitative EEG methodology. Now, EEG has actually been around for almost exactly 100 years now. The first EEG was performed by Hans Berger in July of 1924. We collect millions of EEGs every year, and yet we have not extensively examined these signals. We've all seen firsthand the power that AI and machine learning can do, but for some reason, we've largely been ignoring this rich data source. So here we see our cohorts. Each dot is one EEG recording. And again, these are all retrospectively collected EEGs. The x-axis is the age of that recording. We also see on the y-axis the total number of EEGs and the total number of individuals in each cohort. So you'll see with KCNT1 currently from retrospective EEGs collected at CHOP, we have 12 individuals with 65 EEGs. And here you can see it's actually a pretty nice spread across childhood. This is a pretty rich starting point, especially for such a rare genetic disorder. And so we can analyze the changes across childhood without significant gaps in sampling until we get to late teenage years. We also have isolated nearly 1,000 EEGs from controls with normal EEGs. And if you're interested, I can go into the really in depth what we did to isolate these controls. It wasn't a straightforward process, but now we have this rich data source to compare to populations like KCNT1. Now, here you can see that nearly our entire KCNT1 cohort of individuals are diagnosed with epilepsy of infancy with migrating focal seizures. However, we do have one individual with sleep hypermotor epilepsy with 5 EEGs. But we excluded this one individual from most analyses unless otherwise stated to prevent potential confounds. But of course, when attempting to isolate unique biomarkers from a condition like KCNT1, it's not enough to compare to neurotypical individuals. So in my analyses, I also have access to thousands of other EEGs from individuals with other developmental and epileptic encephalopathies. Here you can see some other genetic conditions, SCN1A, SYNGAP1, each a different DEE. You'll see later that I can leverage this resource to help isolate unique signatures in KCNT1. Now, this is one of the largest cohorts of childhood epilepsy EEGs ever gathered and analyzed. I'm not even including the other 80,000 EEGs we've downloaded from a variety of different conditions. Now, taking this a step further, we can also try to isolate biomarkers of severity and outcome with EEG. Individuals with KCNT1 often struggle with motor development and motor control. Now, the GMFM, or Generalized Motor Function Measure, is a motor test that measures this. These are dots from EEGs from individuals with a GMFM test completed close in time. Or on the bottom, as you see, a seizure frequency noted by a clinician close in time to EEG. I've been analyzing also the ability of EEG to predict and track motor development and things like seizure control, which could be incredibly impactful when trying to objectively measure a child's development or the effects during a clinical trial. Now, this is a bit beyond the scope of my talk today, but if there's time and interest, I can present these results as well, or you can also check out my preprint. All right, so here we see what I showed earlier with the addition of an artifact rejection algorithm. In orange, you'll see it isolate and remove a clip that contained noise. It's essential as a variety of things can happen during an EEG. Excess noise, a child scratching their head, etc. So we need to be able to automatically identify clean epochs of EEG data. We can then use the location of the electrodes. The right is where the electrodes are placed. And if you'll recall, each of those squiggly lines is a single electrode. Now we can average signals from electrodes in certain regions of the skull, and then we can extract information from different aspects of the signal. Now, this is what's called a power spectral density curve. All I want you to gather is that we are essentially deconstructing the signal on the left into different components that we can better analyze and use for quantitative EEG or qEEG. Now, each one of those lines is one EEG from a control patient and the extracted power at different frequencies. So this is almost 1,000 EEGs across ages of 3 months all the way to we actually have a 30-year-old. And we can see how it shifts across development. This is a constantly changing signal as a child develops. Now, let's take a look at one particular frequency band called alpha, which is shaded here in red. Now, this is a relatively simple quantitative EEG measure that we can just look straight off the bat. This is relative alpha power across childhood and the nearly 1,000 neurotypical individuals. The y-axis is this relative alpha power across electrodes, and the x-axis is age in years. Each dot is an EEG, and the shaded region is an approximated 95% confidence interval. Now, what does it look like in an individual with a KCNT1 related epilepsy? Right off the bat, we see a clear and striking difference. Now, there is variability, but a clear depression in relative alpha activity across the brain and across development. But this is against neurotypical individuals. What about another DEE? Is this also unique to KCNT1 or is it common? Well, to begin, let's look at a disorder in which we have a bit of higher sampling. Here you see individuals with SCN1A-related epilepsy. Reduced, but not as reduced as KCNT1. There's still a clear and striking difference. However, some variability. I also want to note the clear difference at certain ages, and there's a difference in the trajectory. Now, using my quantitative EEG pipeline and a substantial cohort size, I can almost entirely automate this process. Next, we wanted to focus on a particular biomarker that has shown promise in other diseases. It's often a mark of severity, also a rough estimate of sleep. This is called the alpha-delta ratio. You can sort of think of it as an estimate of slowing in the brain. A lower alpha-delta could indicate an individual is more affected by the disease. So here we group them by age groups. You can see the different colors indicating a different cohort. Red is controls, turquoise is SCN1A, and purple is KCNT1. Displayed significance is after a multiple testing correction. 3 stars is indicates a p-value of less than 0.001 after, again, multiple testing. In infancy, we see significantly lower alpha-delta ratio in KCNT1, or increased slowing. We see an even more striking difference in toddlers, similar in children and in adults. And together, the median difference between controls and the KCNT1 population across the age span seen here achieves Cohen's kappa of about 0.9. So that's sort of an effect, you can think of it sort of as an effect size. But then in context, a very successful drug might have a Cohen's kappa of 0.6. Anything above 0.8 is very robust result. Next, we wanted to see if we could use AI and machine learning to see if they could predict a KCNT1 from controls. Now, this is not a straightforward task. All the model is given is EEG features, and each individual with KCNT1 has an age and sex match control, and then we perform what's called leave one out cross-validation. We tested 2 models. One model is given spatial information about which area of the head the signal is coming from. In other words, Are they coming from frontal electrodes or occipital electrodes? The other model has features averaged across the skull. No spatial information is given. From this, we can test if their abnormalities are at all focal. So again, the utility here is not necessarily predicting the disorder, rather using advanced techniques to quantify electrophysiological difference and potentially isolate novel biomarkers. Here we see the performance in KCNT1 against age and sex match controls using what's called an ROC curve. So as that bow-like shape reaches closer to the 1, that's a better and better performance. And we can measure what's called the area under the curve, the AUC. Now, a value of 0.7 is considered pretty good, especially for a complex problem. You'll see two lines. The red line is when we don't give the model any information where the electrode is coming from. The upper purple line indicates we gave the model spatial information. It knows if the signal came from the front or the back of the head. Now, a couple notable results. First of all, an incredibly high performance. I triple checked these results and checked them again this morning. In all my research, I've actually never achieved an AUC of 0.95, so that was quite exciting. Secondly, as you'll see, there's almost no difference. You can run a model, a test, and there's no significant difference between these models and their performance. Now, the fact that we don't see a difference between a spatial and the non-spatial model in performance, this could mean one of two things. First, the electrophysiological abnormalities seen in most individuals with KCNT1 are non-focal and completely generalized. Or, which is, I think, the much more likely option, they have some focality, but the abnormalities are at least somewhat generalized, and the differences are great enough that the model doesn't even need to know where the abnormalities are most focused. Now, we can also get information on what features the models find most useful. What differentiates these populations from controls? And I should note, this is done in a nonlinear fashion. On the y-axis is feature importance, as measured by something called the Gini index. The abbreviations at the end of the features on the x-axis, such as CNT and PAR, are shorthand for lobes. So these are either average across the central lobe, parietal lobe, frontal lobe, etc. And as you see, the spatial model found that the alpha, theta, and the central and parietal lobes best differentiated KCNT1 from controls. Now, to help answer the focality question, or at least provide some evidence in one direction or the other, I trained another model with both these focal and these diffuse features. Then it can tell us in a similar way what features it found most important. Well, the model performed about just as well, an AUC of 0.94. And the all-feature model, interestingly, still mostly shows the same focal features. You'll see even the top 3 most important features were the same. So what does alpha, theta, and the central lobe look like in our cohort? Well, you can see why the model chose this feature. Now, there's one outlier. I don't know if you can see my pointer. But beyond that, there's a pretty clear, consistent difference. Now, if you'll recall, we have one individual with sleep hypermotor epilepsy, and I actually included them in the machine learning model that performed so well. Do they look different here? Is this particular biomarker specific to just epilepsy of infancy with migrating focal seizures, or is it applicable to all KCNT1 related epilepsies? Here we see that individual, highly variable, but for the most part, they are not an extreme outlier. Now, again, this is a sample of 1, so I'm not going to claim or presume anything, but this is a notable result. Now, some people will fairly state this high performance is simply because we are comparing to a healthy population of individuals. It's not necessarily a fair comparison. Are these signatures unique to KCNT1 or just DEEs or simply more affected individuals with a genetic epilepsy? Now, I can tell you right off the bat, this is the highest performance model I've seen in individuals with a particular genetic etiology. Nevertheless, to take things to the next step, we need to be able to compare KCNT1 and their EEGs to other DEEs. So I tested a three-way machine learning model. This is, again, a random forest model that had to predict if an EEG originated from an individual with KCNT1, SCN1A, or STXBP1. Again, not an easy task. We're given other DEEs, which I'm sure have some sort of overlap in their electrophysiological signal. And it's having to decide between three different outcomes as opposed to the binary control versus KCNT1. Nevertheless, after running 1,000 permutations, this is how well the model performed. This is a form of what's called a confusion matrix. On the y-axis, you'll see what is presented to the model. And on the x-axis, you'll see what the model actually predicted. And so the diagonal is where the model correctly identifies where the EEG is coming from. So in the middle of each box is the mean accuracy across permutations, and the brackets is the 95% confidence interval. So it performed extremely well despite the complexity of the task, achieving almost a 90% accuracy in KCNT1. And interestingly enough, as backed by our previous models, it performed best at differentiating KCNT1. And another thing to note that I find interesting is that it almost equally mischaracterized individuals as STXBP1 or SCN1A when presented with a KCNT1 individual. Both had an error rate of about 5%. Oh, here we go. So you'll see that the model performed extremely well. Yeah, 90% accuracy. Now, one thing I do want to keep in mind, however, is that these patients are not age matched, as the sampling of when these child received the EEG can be extremely different. So I also trained models that were only given age of the EEG as a feature. This achieved about a 70% accuracy. So the unique EEG signature of KCNT1 was essential to achieving this high level of accuracy. Now, next, I wanted to see what does this performance look like when we make it a binary prediction model, KCNT1 versus other STXBP1 or SCN1A, or SCN1A versus STXBP1 and KCNT1 together. This is far easier for the model, and thus we should see an even higher boost in performance. And we do, again, incredibly high performance. I checked this again many times, and I was hesitant to show it to you because I doubted, because the performance was so high. And this time I removed age as a feature completely to try to mitigate the bias of age, as the age distribution of when these EEGs were taken varied by disorder. Nonetheless, I want to make sure I get the point across that one must keep in mind that age still may play an underlying role, as these EEG biomarkers change across development, though in perhaps unique ways for each disorder. So what does the model tell us about the most important feature in this three-way model? Here we see the important features and how age helped the model. However, again, we see the importance of the alpha theta. So in summary, we've seen how KCNT1 appears to have a unique electrophysiology, and we can use signal processing and machine learning to extract the features that help us identify this difference from both neurotypical individuals and other epilepsies with striking accuracy. So what are the next steps? What is the future of qEEG? I'm lucky enough to be involved in both the academic and industry side of this question, and I wanted to leave you all with some of my thoughts. So we'll begin to be able to implement these EEG biomarkers and algorithms to provide some personalized medicine in some individuals with certain disorders and abnormalities. Now, it's yet to be seen how far-reaching this will be, how many syndromes and how many diseases or how many medications this may aid. But to give a real-life example, this is a 3.5-year-old with KCNT1 and their first EEG we have on them from CHOP. And this is their alpha theta power. We could see this as their first baseline measurement. But over time, we can begin to track their progress. Perhaps they just began a new medication or are part of a new genetic therapy trial. A simple EEG every 6 months, and we can potentially extract deep insight into their progress beyond simple and often flawed measurements like seizure diaries. Now, we also saw this bump. This could have been random, or something else could be going on. Perhaps a new medication was tried and failed. We don't know yet. But then we see it quickly drops again. Now, how does this compare to a typically developing child? Here we see again the nearly 1,000 control EEGs in red. Now, perhaps we should be looking for treatment responses that result in EEG biomarkers that draw closer to that red line. It's too early to say. Nevertheless, all these purple data points from this child are all potential indications for disease progression at our fingertips that we may be missing. Millions of EEGs are just sitting in hospital data warehouses. Now, what I've been presenting here is classically referred to as qEEG, but there's been remarkable advancements in the past couple decades in an area called network neuroscience, where we can try to measure the connections in the brain, or in this case, electrodes. Now, I've had some very promising preliminary results using network neuroscience for precision medicine. It's still being validated, but hopefully sometime soon I'll be able to discuss it. Now, here we see what's called the median coherence, or essentially connection strength between electrodes in the theta band in toddlers from our control population, toddlers from our KCNT1 population. And visually, we can already start to see some differences. Now, I won't go on to some of my later results in this area, but I just want to give you a visual and some idea of the potential that we see here beyond traditional qEEG measures. Lastly, I want to mention data sharing. To train these models, and especially if we want to train more advanced deep learning models, we need lots of data. The progress in this field will largely lie in our ability to safely and easily share data like EEGs and much more. We can't just rely on EEGs, we need the outcome measures as well. Now, this sort of data sharing will likely involve consortiums and agreements between hospitals, institutions, and companies. This involves trust between these groups and between these groups and the patients. So in conclusion, as we've seen, EEG contains some correlates of disease. And KCNT1 has a distinct electrographic signature that surpasses all of the other genes I've tested thus far. And quantitative EEG can potentially track disease progression across development, particularly when combining with multimodal data like that seen from the medical record and genetic data. The potential is extremely high. I see tremendous promise in this field, and I think we're going to see some remarkable developments just in the next couple of years. Thank you for all of your support. I'm happy to present some other results, but for now, here's my contact information, and please let me know if you have any questions.
Thank you so much, Peter. That was great. If people want to come off mute and ask some questions, I assume that you can come off mute. The floor is open. Yeah.
This is a type of analysis I've wanted to do, but haven't had the team or capacity to do it. So congratulations. This is a lot of work, I can see, but it really lays out the case to use EEG as a biomarker to track, disease progression, but also potentially even treatment response.
That's the hope.
Yeah.
Go ahead, Catherine.
Yeah, thank you so much for that beautiful data. I'm Catherine from SINGAP Research Fund, and Ali generously invited me. I was wondering, how long does the EEG need to be in order to do this kind of analysis? Like what kind of time frame are you looking at? You showed some individual vertical slices that were very short, but I'm wondering what kind of time, what's the length of time you need, and when, and then a separate but related issue is like, how many electrodes do you think you need? And do you think you can get away with the clinical EEGs rather than the qEEGs?
Yeah, Jill and I should mention also Jill McKee is working on qEEG with me actively on SYNGAP1. But both very good questions and some open to debate. But for my paper, we required a minimum of 1 minute of clean EEG. So it really depends on the feature and it depends on how clean the EEG. As you know, not all children will tolerate an EEG. So some EEGs I have to throw out completely. They might have had an hour-long EEG, but simply none of it was usable, or only 30 seconds was. But
And
many of my
can you tell kind of by like behavioral things that are going on, or is that can you only tell after the fact?
I personally have never been in the room during a long EEG session, so it's hard to say. But things like scratching the head, violent moving, a tech will be able to tell by looking at the signal how much they're actually gathering. I've been in conversations where, oh, hey, Peter, I think we just got a new SYNGAP1 EEG, but you probably won't be able to use any of it. I've heard things like that once before. So ideally, you want the child as still as possible, but some movement is okay, and we can try to control for that. But many of my EEGs are, 22 minutes long, and I extract 2 minutes of data. There are more complex features I'm using where we need more like 5 minutes of clean data, of continuous clean data, which is a high bar to reach. But for these qEEG measures, I would say you'd want 1 to 2 minutes of clean data. As far as the number of electrodes, um, it's still an open question, but I will say one of the biggest impediments, I mean, I shouldn't say that. A big issue in this field is when we're using electrode devices and systems from different hospital systems that use a different format. So what's honestly, you might be surprised to hear, the easiest for me to deal with currently is if everyone just uses the standard clinical 1020 system. You can gather more data from complex systems, but if you want to analyze a massive cohort of retrospective data or just make things very easy, just using the standard 1020 system. In the future, especially with what we're picking up, the importance of certain electrodes, I'm sure that you can still grab some remarkable data from just a few well-placed electrodes. But what I would tell people just as a baseline, a standard clinical scalp EEG that with the 1020 format is great. And as long as the child's staying as still as possible, if you can get a couple of minutes of clean data, that's what we shoot for now. More is better, but.
Peter, and it might just be just worth pointing out that basically, I mean, we have good validation from our clinically acquired EEGs and our quantitative EEGs. So this is kind of whatever we do in terms of EEGs is actually sufficient for you to extract the features.
Yes.
That's great. Thank you. I'll let Christine go. I have a few more questions, but I'll let somebody else go.
No, that's okay. Peter, probably the first time we're meeting. I'm Christine Kogrim from Ionis Pharmaceuticals. So it's a pleasure to meet you, a pleasure to meet many of you probably for the first time. Thank you so much. Very, very informative. We're very interested in electrophysiology as biomarkers across all of our programs. And just maybe a couple of follow-up questions. One was related to what kind are these research-grade EEGs or clinical EEGs? And then just a little bit of an understanding about kind of the standardization. Are you looking for these are awake, both eyes open, that sort of thing? I'm assuming that's the case.
Yeah, so sorry, can you repeat the first question?
The question was just about like the standardization of how you're capturing these EEGs. So I'm thinking about both eyes open, awake state.
Okay.
Here.
Yeah, so for our first pass, we wanted to do something. We wanted to try to capture resting state. So not asleep. We removed any indications where they were asleep, seizures, any sort of photostimulation. We wanted eyes open, though, of course, it's hard to control when you're trying to analyze thousands of EEGs, but we're going for resting state. However, I will say that should just be the beginning. There's a lot of evidence that some disorders, you might only see the most prominent abnormalities in sleep. It really depends on the disorder. And I think you're going to pick up specific signatures that may be more informative when their eyes are closed, et cetera. But for this first pass, and what's actually shocking is that we still see a very clear signal in just resting state across these retrospective clinical EEGs. And again, as Ingo stated, we're validating it in these research EEGs as well. But there will be probably very informative biomarkers, even with things like people are experimenting with transmagnetic stimulation in EEG for getting a unique signature for certain diseases. The field's wide open, but I would say you can still get particularly in these disorders, a lot of bang for your buck and just resting state.
Resting state, yeah, and to follow up with just a quick question, and maybe it's out of scope for this, just thinking about evoked potentials. So, are you guys doing any work on evoked potentials, auditory or visual evoked potentials in this population? Do you have an interest in doing that? So again, just try to amplify that difference and really capture something that you feel is a true biomarker.
Yeah, absolutely. We will be doing it in our ENDD clinic. So we have that capacity, but currently these are all retrospective scalp EEGs. I mean, that's where we're going to get our high numbers.
Right.
But I will add that there are one thing I do want to analyze. Ingo, if you can get me a PhD student, I want to get, or a master's student, I want to analyze a lot of these children have evoked potentials in some of their retrospective, these clinical EEGs. And those are marked. And I have ways to extract that. So that is something on my list I'd like to look at. So absolutely. And we are looking at it prospectively in our research EEGs.
Yeah, Peter, you'll get your PhD student, I promise. And Christine, there's a different EEG setup from the retrospective area that Peter has worked on, and we have this for KCNT1, where we don't have an ongoing natural history project. Yeah, for other conditions, we actually do prospective quantitative EEGs, and then we're going to face in evoke potentials too. Yeah? It's not that problem because we have good biomarkers for some of the conditions we work on. So that's not the main thing on our kind of focus right now, but we'll get there.
Great, thanks.
And I want to note someone, Katie, by clean data, do you mean signal that is free from interictal discharges? What about seizures? Do you try to take a sample with X amount of time away from seizures? Great question. So yes, most of the seizures are annotated, and we remove chunks of time before and after and remove the seizure from the EEG, as all these EEGs should be clinically annotated. As opposed to the interictal discharges, no, that's a great question. Those we don't. I have a friend who's running what's called SpikeNet from Brendan Westover's lab, and they're using that in their scalp EEGs, but we haven't run it yet on this data. So no, we aren't accounting for interictal discharges, but it's something that absolutely does need to be taken account of. And sorry, I know I can't keep. Someone can open their microphone. I think I see a hand. Catherine?
Oh, yeah, thank you. So I wanted to ask you about your slide where you showed the interconnectivity. There were kind of two bright yellow spots in the KCNT1 side that weren't in the control side. Mm-hmm. And if I remember right, the bright yellow was the less connectivity.
Yeah, I would say that this is all very, I wouldn't try to
okay,
going forward.
We can stop my question then. I was wondering if that would be a lead. Let's not call it a biomarker. Is that like a lead for a potential biomarker if you see regions of the brain that don't connect as well with each other in the patients?
Yeah, sorry, I thought I was sharing it. So if that didn't make sense what I was saying before, but yes, so it could be, it's all potential, yes. I hesitate when there's a single electrode pair, just because of the variability and the noisiness of the signal.
So they could have had less contact or something.
Yeah, I mean, this was the median taken across toddlers, but I need to dig into the network measures more. I've actually looked at coherence less, and I've looked at other entropy-based measures, which have shown a lot of promise. But yeah, absolutely. I think the network features could show a tremendous amount of promise. And also looking at...
I noticed on the KCNT1 side, it wasn't plural. It was like toddler instead of toddlers, but that's a mean for multiple people.
Yeah, it's a toddler. Yeah, it's sometimes I designate group toddler. Yeah, it should have been toddlers. Yeah.
Okay. Yeah, that part was fascinating to me. And then when Christine was talking about the evoked potential, I was thinking for SYNGAP anyway, one thing that is really different about SYNGAP is not the reflexive seizures, because I know you don't want seizures, but the like when my child, he's got this really great visual memory, right? And then he like has this addiction cycle of wanting something. And so when he watches a certain part of a movie or hears the song from the, that's the layover of that soundtrack from that part, he has like a very different behavior. And I always wonder what's going on in his brain. And I wonder if when he's watching and when he's listening are the same. So anyway, that sounds more like fMRI to me, but I don't know if Ingo's laughing.
Yeah. No, you.
I'm just trying to kind of see. So, Catherine is maybe off topic. But we have like our SYNGAP biomarkers, which is basically, so Peter has seen like in KCNT1 we have this centrally. For SYNGAP it's more occipitally where we have much of the kind of visual processing. And I think this is things where we just kind of see whether this validates in the SYNGAP population.
Yeah, well, I was thinking what Christine was saying. She was talking about evoked, like what would you evoke, right? Like what is special about every single, not just SYNGAP, but any population, what's special about them? And then where do you see it more strongly? And then that might be a place you could see the change more strongly when you're getting corrected with a therapeutic.
Yeah, and so Catherine, those would be VEPs or visually evoked potentials for SYNGAP. This goes directly into the occipital cortex.
Okay, and those aren't the same as seizures, because you said you didn't want to look at seizures. Those are different.
It's almost like a reflex of the brain once you kind of are exposed to a light.
I see. Okay.
Do SYNGAP patients, are most of them aware enough to focus on like a checkerboard pattern? You could get a stronger visual evoke with a checkerboard than you could with a flash.
I think we should maybe ask Ali first if we're okay to talk about SYNGAP in the KCNT1 meeting.
No, never mind. Because I know KCNT1, like EMFS patients, are probably not going to work, but they don't have the occipital signal anyway. So then I'm going to think about what central or parietal evokes you could do.
So there's probably going to be a SYNGAP qEEG seminar soon. Yep.
But hi, I'm Sarah Ruggiero. I'm also associated with this research group. So visual evoke potentials have been successfully used in CDKL5 previously, which is those children are about as developmentally impaired as individuals with KCNT1. It is that checkerboard pattern, and they are able to focus on it. So it is a potential to be used in this population, likely. Just wanted to weigh in there.
There's some interesting work in autism as well.
Yeah.
Yeah, and that's what I was going to clarify. We work on a lot of neurodevelopmental disorders, including developmental epileptic encephalopathies, and it's based off where we think the damage is in the brain and which pathways are impacted. That you might select, an oddball tone for a mismatched negativity if you think there's brainstem involvement and you're looking for, an auditory evoked potential versus some sort of visual. And I agree with Viet, if you can get, focusing and you believe that there are regions of the brain that are impacted, you can do the checkerboard, or you can do a light flash, where we do lots of those types of things across our trials, but it really depends on where you think you're going to see the signal based off of what you believe to be the processes involved in the brain.
Anyone else have a question?
Yes, it's Greg Kazarowski. I joined late, but my question is, how do different drug cocktails affect your results that the patients are on?
That's a great question. And we can't control for it. I will say, however, that I've started to do some more work on the effects of ASMs. And I will say that the literature actually is quite muddled in how much it affects the spectral features. There's a few recent papers out on this. And so with certain medications that are more affected, like certain benzos, you can see more of an effect in the literature, but a lot of literature actually struggles to use qEEG metrics. I want to emphasize qEEG metrics. I think there are other metrics that you can start to see, and I have some preliminary evidence showing that you can sort of start to see how ASMs affect other aspects of the EEG, but there's inconsistent evidence in how it affects the spectral features. I think it does, but in a substantial way, it hasn't been shown except for in certain medications.
You really can't wash out the medications here, right? That wouldn't be ethical. So, and I would think there'd be some variability induced by the cocktail.
Oh, no,
aspect?
absolutely. Absolutely. On an individual medication basis, besides, I would say, for like benzos, the literature is pretty inconsistent on having an extremely strong effect for spectral features. But absolutely, it is having an effect.
Yeah. Peter, I would even be stronger. I would say basically the fact that we have so many patients on so many different medications and combinations of medication basically averages this out, and the features you're seeing, they're so strong that they're beyond this. Huh? Plus, we have a very different variety of medication combinations in the control group. Yeah? So I think what we see as a signal cannot be attributed to medication effect.
I would certainly hope, though, if you had a therapeutic that you thought was effective, you see a marked change.
Or a disease modifying treatment.
Right.
Yeah.
Either one.
Oh, I see another question in the chat. Since you have multiple EEGs for patients over time, have you been able to look at trajectories in these qEEG signals in individuals over time? Wondering how stable these are. Relatedly, any prelim data, how the signals you're measuring relate to important clinical factors as seizure severity. So no, we have not done I would say I haven't done focused work on individuals and their trajectory. It's quite difficult when you're trying to take into account a variety of the different things. I've attempted it with certain individuals where I have 20 EEGs. And it's quite hard going through the medical chart, and a lot of these are very complex patients, and trying to attribute one thing to the other. You need consistent measurements, like those things going on at ENDD, where they're getting an EEG every 6 months with a battery of other tests. It requires a lot of work, but I think that's the future. I mean, if you want to take a more precision medicine approach, or if you're doing a clinical trial, then you want to keep into account absolutely everything that's going on and get consistent EEGs in the same setting. Prelim data about the signals that relate to clinical factors.
And to Peter, maybe just to kind of comment, I mean, we've done this on a few patients that were, let's say, outliers, where you actually followed this through and could show that the quantitative EEG features were constant over time. I mean, that's the assumption. So we don't expect them to kind of vary. We don't expect them to respond to medications, but we think on them as a inherent feature, as a biomarker of the disease, and not so much as some kind of like a meta phenomenon that is easily influenced by medication or seizure severity. So it's quite close to the underlying biology.
Yeah. And the seizure severity question, my friend wrote a paper on that in intracranial EEG. You can check that out. We have not. It's hard to measure seizure severity. I've attempted to predict seizure frequency across patients, and that's in my preprint, and we showed that we could not. But I do have good data showing that there are predictive elements in qEEG to predict motor scores. They can't predict that they won't replace the test themselves, but there are spectral features that help predict, would provide some information pertinent to motor development. Epilepsy severity. Yeah, it's difficult. We need to measures like consistent measurements like the GMFM, which I mentioned, which I'm happy to talk about another time.
Okay, so I've got maybe a really practical question. In thinking about decentralized trials, which we hope are the future for KCNT1 individuals, we are hoping to do home EEGs for everybody and try to convince everyone that that's what needs to happen for our population, since they're so fragile. Do you have recommendations about how to do those home EEGs? What helps us get the best possible, cleanest data, if you have any experience with that?
I would refer to a clinician. I don't want to presume on this one, besides saying, beyond the basics of them not scratching their head and jumping up and down. But I'll defer to Ingo on this one.
I have to differ further. I would just say typically when we do like ambulatory EEGs, they work quite well. It depends a bit on how you institute the EEGs and where they're being done. If those are ambulatory EEGs or EEGs that kind of families would use them themselves. I think it's a bit of trial and error with the specific system that you're using.
Peter, with the CHOP data set, is this predominantly outpatient EEGs or some of these inpatient ones?
Yeah, some are inpatient. And when we move forward with something like with my the paper, we made sure that these weren't done in a emergency setting. So they could be inpatient, but we made sure that, first of all, we only took the beginning segment. They weren't asleep, but most were outpatient. But we try to gather all of them. We want to get all the data we can and try to mitigate for things like, is the child having an event of status or in an abnormal state? So we're trying to capture how the child is doing typically.
So there, you didn't have records where they were in the ICU and not awake. You mentioned you were trying to capture the awake resting state. And so did you have records in which there was no awake resting state throughout the record because they were in the ICU?
For this data set, I don't recall. There were, in my paper, I did remove, I want to say 4 or 5 EEGs that were done in an emergency setting where I went through and read their charts. Because sometimes it's not very interesting. Sometimes it's labeled as EMU, but it's not really an emergency setting. And sometimes, so it requires manual chart review. So yeah, we did try to read through and make sure that these weren't done in an emergency setting. In reference to them being entirely asleep, I'm not sure. I'd have to go and look back at my notes.
Yeah, a quick question. I'm sorry I joined late, but I'm hoping, is your paper available to read and we can get access to it to help supplement what we've learned today?
Yeah, absolutely. I will say it's the preprint, so the results are a bit better and the paper coming out, but yeah, absolutely. It is available on medRxiv. I could try to pull up the link very fast and put it in the chat.
Yeah, I can put this in the chat quickly. You can kind of take a few more questions. Then you don't need to. I already got this.
And I'm sorry, maybe I missed this at the beginning, but how did you establish your patient population for this study?
For the paper?
Yeah.
Yeah, so the paper, we wanted to focus first on 3 other genetic disorders, SYNGAP1, SCN1A, and STXBP1. And so we gathered all of those individuals that we have records of at CHOP. Most of these have been recruited through different initiatives called ENGINE or EGRP. But the control population, we go through in the paper, but essentially we wanted to ensure all EEGs were marked as normal by a clinician, and we went through a long list with 3 different clinicians of any potential diagnosis code that could alter the EEG, ranging from things like schizophrenia, bipolar, to I think I may have even removed ADHD patients. So we tried to be very thorough, and they had to have at least a year of follow-up. So these are marked as normal by clinician, and they don't have any evidence of any diagnosis of a condition that could impact EEG. But, as best as we could, we tried to control for those factors.
And I know there's some other public EEG data sets have been available for a long time, like the Temple University corpus and things. And I'm curious if you used any of the biomarkers that you've discovered in your population and seen if they exist in some of those other data sets, although I know they're not labeled with genetic diagnoses. But I'm just curious if you've been able to perhaps find, like a patient from the target population in one of the other data sets.
No, we haven't. We wanted to try to keep this as clean as possible, so a data set entirely within CHOP, and we had the population. It would be interesting to look at, but there are just so many additional variables when trying to combine especially data sets that's older and from a different hospital system.
Yep.
It would require a lot of work to look into it. And for our first paper also, we just wanted to present as clean results as possible because, there's been a lot of doubt that scalp EEG can hold any potential. So we wanted resting state, awake, keep things simple and clear. Yeah. But yeah, it'd be interesting to look at. I know that some of those data sets exist and more should.
And so Peter, if I remember this correctly, this is not pediatric data, right? This is largely
That's true. Yeah.
Data. Yeah. So I think when it comes to pediatric data, Kevin, there are actually very few resources, and Peter has really built a very nice pipeline that gives us access to all our EEGs. Yeah?
Right.
Okay, so I'm guessing this hasn't been done yet, but have you taken any of your algorithms trained to pick out KCNT1 individuals or some of the other DE conditions and then run it back on some of your data, maybe from kids that are starting to come in that haven't been tested yet, to find out, can you pull them out before you have your answers?
No, I haven't. No, I have not, but that would be interesting. But luckily, I mean, bad for this research question, but good for the patients. I mean, CHOP does, I mean, Ingo could answer this better. They're going to get genetic testing hopefully fairly quickly. But we did something similar in another paper to see if we could identify features from their chart to predict the disease. But the EEG, we have not looked at that, no. But it would be cool to see.
Thank you so much, Peter. This has been really wonderful. We really appreciate you coming to give this seminar and answering all these questions. We are going to put this recording on our YouTube probably for a short period of time. So if any of your colleagues miss out on this, we will send out a link eventually once we get this cleaned up and ready. Thanks again, Peter. This has been great. And thanks everyone for showing up today. Have a great day.